Hello students, welcome to E-Pathashala. I am Dr. M. Pungodai, working as Assistant Professor in Zoology, Avanashalingam Institute for Home Science and Higher Education for Women, Coimbatore. Let us have a glimpse on the motility and hormones of gastrointestinal tract. This module helps to know the changes that occur when the ingested meals pass through the gastrointestinal tract, promoting numerous endocrine and exocrine secretions which digest complex molecules present in the meal into simpler ones and absorbed by the epithelial cells. During this process, mixing, turning over and propulsion of the luminal contents occurs in the organs of gastrointestinal tract, which also releases a group of hormones from the specialized cells of the digestive organs to carry out and control various functions. The objectives of this module is to enable the learner to understand the intensity of mixing and rates of propulsion of ingested food materials which differ in various parts of the digestive organs, to know the types of contraction that plays a diverse role in gut motility function, and also to know the role of gastrointestinal hormones in digestive process. What is gastrointestinal motility? Gastrointestinal motility is a movement of digestive system and the transport of food materials within it. The food materials taken through mouth is passed through the alimentary canal and then digested into smaller particles. During this process, a number of movements takes place in the alimentary canal, namely mastication, deglutition, gastric motility, motility of small intestine and large intestine, and the defecation. Mastication or chewing is a mechanical process where the food particles are broken down into smaller ones with the help of saliva, which act as a lubricating and moistening agent. The thoroughly crushed and grounded spherical soft balls called the bolus are pushed backwards for swallowing. During mastication, movements such as opening and closure of mouth, rotational, protraction and retraction of jaw are involved with the help of masseter, temporal, pterygoid and buccinator muscles. Mastication is the reflex process which is carried out voluntarily, and the center for mastication is located in medulla and cerebral cortex. Deglutition or swallowing is a process by which the food is passed from mouth to stomach in three phases, namely the oral phase, pharyngeal phase and esophageal phase. In oral phase, the bolus formed in the mouth is pushed by the tongue against the hard palate, forcing it towards the posterior part of the mouth and into oral pharynx by voluntary action. The pharyngeal phase is a reflex phase which is initiated when the bolus triggers the tactile receptors in oropharynx. The passage between the nasopharynx and oropharynx is collected when the soft palate is elevated. The larynx rises inside the neck and the epiglottis moves to cover it, providing even more airway protection. To receive the bolus from the mouth, pharynx gets elevated. The pharyngeal constrictor muscles then contracts the succession, forcing the bolus to pass through the pharynx. Parallelly, the upper esophageal sphincter relaxes and the food is pushed into the esophagus. In the esophageal phase, the food enters the stomach from the pharynx with the muscular contraction called peristalsis, which occurs in the walls of esophagus. The circular esophageal muscles allow expanding of the digestive tract when bolus is propelled through esophagus. The lower esophageal sphincter relaxes as the peristaltic movement approaches the stomach. The presence of food in the esophagus activates enteric plexus, which controls peristalsis through local reflexes. The motor impulses passes through the efferent fibers of vagus nerves to the muscles with esophagus, which stimulates their peristaltic contractions. During gastric motility, the food materials enter the fundus of stomach and with the help of stronger peristaltic waves, they are passed down to the antrum of the stomach. With the help of segmental contractions, the food gets thoroughly mixed and the mixed fluid contents known as chyme is pushed towards the pyloric sphincter into the duodenum with the strong peristaltic contractions. The peristaltic wave passes through the partially opened pyloric sphincter, which causes the chyme to move in a back and forward fashion and break the larger molecules or materials left in the chyme. Increase in stomach motility promotes stomach emptying. The rate of emptying of stomach is controlled by diverse factors originating in stomach and the duodenum, of which the duodenal factor seems to be important. The increased volume of food in the stomach and stretching of the stomach wall is initiated by gastric factors. The hormone gastrin promotes stomach emptying, and to inhibit the sudden influx of acidic chyme into small intestine, duodenal factors play a very important role or the vital role. Presence of chyme in the duodenum creates nerve reflexes which are transmitted to the stomach by the enteric nervous system and through extrinsic nerves via the prevertebral sympathetic ganglia, and thereby inhibits gastric emptying. Distension of the duodenum, degree of acidity of the duodenal chyme, osmolarity of chyme, and irritation of the duodenal mucosa are the main factors that inhibit gastric emptying. The hormones that inhibit gastric motility and emptying are secretin, cholecystokinin, vasoactive intestinal polypeptide, somatostatin, peptide YY, and gastric inhibitor peptide, which are secreted in response to acidity in the duodenum and presence of fat in the chyme respectively. The motility of small intestine is vital for mixing the chyme with digestive juices, propulsion of food and absorption. The food is slowly passed through the small intestine by segmental contractions and peristaltic contractions through the regulation of local reflexes. The chyme moves down the small intestine by sweeping movement at a rate of about 1 centimeter per minute to reach the ileocecal sphincter in 3 to 5 hours. The ileocecal valve, a sphincter, is generally in a constricted condition, but when movement in the ileum increases, this sphincter relaxes and allows the food residues to enter the cecum of large intestine. Both neuronal reflexes and hormones control the relaxation of ileocecal cecal sphincter. Neuronal factors such as distension of intestine wall and stomach will also cause increased small intestine peristalsis. Hormones such as gastrin, cholecystokinin, insulin, motilin, and serotonin increases peristalsis, whereas glucagon and secretin inhibit peristalsis. The digestive movement in the stomach hassles the gastroileal reflex, which increases the force of ileal segmentation, followed by the release of gastrin by the stomach, which enhances ileal motility and thereby relaxes the ileocecal sphincter. The backward pressure helps to close the sphincter, preventing backflow of chyme into the ileum. This is the reason for the complete emptying of food taken at lunch from the stomach and the intestine by the time you eat food for dinner. Migrating motor complex, a type of peristaltic contraction, occurs in the stomach and the small intestine during fasting, helps to sweep the excessive digestive secretions and residual ingested materials into colon and prevents the accumulation of secretions in stomach and small intestine. The intestinal villi also helps in the absorption of digested food particles from the lumen of the intestine by local nerve reflexes and also by the secretion of villikinin, a hormone which increases the movement of villi. When chyme moves from the ileum into cecum, ileocecal sphincter regulates the motility in the large intestine. After the meal, peristalsis in the ileum forces the chyme into cecum. When the cecum is inflated with chyme, the contraction of ileocecal sphincter gets strengthened. Once chyme enters the cecum, three types of prominent movements occurs in the colon of large intestine. The food residues present in the colon stimulates a sluggish segmentation in the transverse and descending colon of large intestine by segmentation or haustral contraction. When haustrum, a small pouch caused by the circulation, is inflated with chyme, its muscle contracts and pushes the residue into next haustrum, which occurs about every 30 minutes. The food contents get mixed in the colon and thereby facilitates the absorption of water and minerals. The next movement in large intestine is peristalsis, which is slower than in proximal portion of the alimentary canal. The third type is Moss movement, which occurs 2 to 3 times a day, usually after meal, by the gastrocolic reflex, which is responsible for the formation of feces and the filling of rectum. When the rectum gets filled, the internal and sphincter gets relaxed and external anal sphincter is under control. The contraction and movement of colon are increased and decreased by the parasympathetic and sympathetic nerve fibers. When the time to defecate is convenient, the inhibition is released and external and anal sphincter under voluntary control gets relaxed and the defecation relax is triggered. In the process of defecation, the large intestine absorbs the water and converts the liquid chyme residues into semi-solid feces or stool, which is composed of undigested food residues, unabsorbed digested substances, millions of bacteria, matured epithelial cells from gastrointestinal mucosa, inorganic salts, and adequate water to allow it pass smoothly out of the body. Of every 500 milliliter of food residue that go through the cecum every day, about 150 milliliter becomes feces. Feces are removed through contractions of rectal muscles. Hence, by the voluntary process called Valsalva's maneuver, the intra-abdominal pressure can be increased by the contraction of the diaphragm and the abdominal wall muscles and closing the glottis. Gastrointestinal hormones are polypeptide hormones secreted by the enteroendocrine cells, which are found in gastrointestinal tract and pancreas with endocrine function. These cells regulate the motility and secretory activities of gastrointestinal tract. Numerous gastrointestinal hormones are present in our system and a few are discussed here. Gastrin, which is a peptide hormone with 34 amino acid residues and is secreted by G cells of pyloric glands of stomach and also by TG cells in stomach, duodenum and jejunum. Gastrin is also secreted by islets of Langerhans in fetus. During the gastric and intestinal phase of gastric secretion, gastrin is released from the stomach. Stimulation of local nerve impulse plexus is created in stomach and small intestine, and the presence of food in stomach and vagovagal reflex during the gastric stage of gastric secretion are the stimulants for gastric secretion. Gastrin stimulates the gastric secretions and motility and promotes the growth of gastric mucosa and stimulates the secretion of pancreatic juice and pancreatic hormone. The next hormone, which is cholecystokinin, is a peptide hormone of 39 amino acid residues, is secreted by eye cells in mucosa of duodenum and jejunum and trace amount in ileum. Chyme, containing the digestive products of fats and proteins, stimulates the secretion of cholecystokinin. Cholecystokinin helps to stimulate the pancreatic secretion with enzymes, contract gallbladder, accelerates secretin activity, increases intestinal motility and endocrinase activity or the secretions, inhibits gastric motility, augments the contraction of pyloric sphincter, induces drug tolerance to opioids and suppresses hunger. Secretin A peptide hormone with 27 amino acid residue is secreted by the S cells of duodenum, jejunum and ileum. It is secreted in an inactive form called prosecretin and converted into secretin by acid chyme. Secretin is also produced by the simulation of digested protein. Secretin stimulates the discharge of watery and alkaline pancreatic secretions, inhibits gastric secretion and duodenal motility, constricts pyloric constriction or sphincter, and increases the potency of cholecystokinin action. The next hormone, which is the stomatostatin, is a peptide hormone, is secreted in two forms, one with 14 amino acid residues and other with 28 amino acid residues. It is present in the hypothalamus, D cells of stomach, upper part of small intestine and in pancreatic islets. Proteins and chyme with the glucose in stomach and small intestine releases the hormone somatostatin. It inhibits the secretion of pancreatic juice, growth and thyroid hormone stimulating hormones, gastric secretion and motility, and other gastrointestinal hormones such as cholecystokinin, gastrin, vasoactive, intestinal and glucose-dependent insulinotropic hormones. Glucagon is a peptide hormone containing 29 amino acid residues. It is secreted by alpha, A and L cells of pancreas, stomach and intestine respectively. Increased amount of fat and protein in the stomach stimulates the secretion of pancreatic glucagon in hypoglycemia. Glucagon increases the blood sugar level. The next hormone, which is nothing but the gastric inhibitory peptide or glucose-dependent insulinotropic hormone, is a peptide hormone formed by 42 amino acid residues, is secreted by K cells in duodenum and jejunum and also by antrum of stomach. It is stimulated when chyme containing glucose and fat enters the duodenum and inhibits the gastric and intestinal motility, stimulates the insulin secretion and increases the fluid and electrolyte secretion from small intestine. Vasoactive intestinal polypeptide, which consists of 28 amino acid residues, is secreted in stomach and small intestine and traces in large intestine. The stimulant for the secretion of vasoactive intestinal polypeptide is acid chyme in stomach and intestine. It stimulates the secretion of insulin and succus intricus with enormous amount of electrolytes and water, dilates peripheral blood vessels, inhibits hydrochloric acid secretion in gastric juices, relaxes intestinal smooth muscles and augments acetylcholine action on salivary gland. Ghrelin or linomorelin is a 28 amino acid residue containing peptide hormone produced by the P or D1 cells lining the fundus of human stomach and pancreas. This hunger hormone is secreted when the stomach is emptied and stops its secretion when it gets stretched. It acts on hypothalamic brain cells to increase hunger, gastric secretion and gastrointestinal motility to get ready for the food intake by the body. Ghrelin promotes the intestinal cell proliferation, stimulates appetite and regulation of energy homeostasis. Ghrelin favors adiposity and contributes to obesity, and it is termed as saginary or fattening peptide. Ghrelin also has protective effect on the cardiovascular system and helps to control the release of insulin. Ghrelin also regulates the glucose metabolism. Motilin, a 22 amino acid residue containing polypeptide, is secreted by the endocrinocytes or the MO cells in the duodenum and jejunum during fasting. When the duodenal pH is alkaline in nature, it stimulates the release of motilin. The main function of motilin is to increase the gastrointestinal motility, stimulate the pepsin production and insulin secretion in glucose-dependent manner. It inhibits the gastric acid secretion and motor activity of stomach. It participates in controlling the pattern of smooth muscle contraction in the upper gastrointestinal tract. Motility, the housekeeper of gut, increases the peristalsis in small intestine and sweeps out the indigested materials. Increased secretion of motilin accelerates the gastric emptying and also increases the release of pancreatic polypeptide and stomatostatin. Substance P is a neuropeptide composed of 11 amino acid residues, acts on neurotransmitter and neuromodulator. It is secreted at the enteric nerve endings in small intestine and pain nerve endings in the brain. It increases the motility of small intestine, increases the local vasodilation and perception of pain sensation. The hormone peptide YY or peptide tyrosine is a 36 amino acid residue containing peptide is secreted by the L cells of ileum and colon in response to feeding. This can be produced by the enzymatic breakdown of crude fish proteins and ingested as a food product. It inhibits gastric motility, increases water and electrolyte absorption of colon, reduces appetite, suppresses the pancreatic secretion, slows the gastric emptying, increases the efficiency of digestion and nutrient absorption after a meal, and inhibits the intestinal motility and also stops the passage of bowel beyond ileum. Glucagon-like peptide 1 is a peptide hormone with 30 amino acid residues which is secreted by intestinal enteroendocrine L cells located in the distal end of ileum and colon, alpha cells of pancreatic islets and also in the brain. The stimulus for the secretion of glucagon-like peptide 1 and 2 is the ingestion of nutrients including glucose, dietary fibers and fatty acids. It increases the insulin secretion from pancreas, inhibits acid secretion and gastric emptying in stomach. It also stimulates somatostatin release, inhibits glucagon secretion, and it is also an anoxygenic peptide which increases satiety, mediator of ileal brake, and reduces gut motility. Glucagon-like peptide 2 is another hormone which consists of 33 amino acid residues containing peptide and is produced by the intestinal endocrine cells and found in brain. The presence of food with the glucose in small intestine stimulates its release and increases the intestinal growth, regulates energy, absorption and integrity of intestine, inhibits gastric acid secretion and will lead to suppress appetite. Glycetinin is a 69 amino acid residue containing peptide generated in the L cells in jejunum and duodenum of intestine, alpha cells of pancreatic islets and also in the brain. Proglucagon, the precursor of glycetinin, is converted into glucagon and glucagon-like polypeptide 1 in pancreas and in the intestine of as glycetinin and glucagon-like polypeptide 2. The biological actions of glycetinin include stimulation of insulin secretion, inhibition of gastric acid secretion, regulation of gut motility and stimulation of gut growth. Glycetinin also increases the blood sugar level and kidney and plays a major role in the clearance of glycetinin from the circulation. Pancreatic polypeptide is a polypeptide consisting of 36 amino acid residues and secreted by PP cells in the islets of Langerhans in pancreas. In human, its secretion gets increased after a protein meal. Fasting, exercise and acute hypoglycemia and decreased by somatostatin and intravenous glucose. The function of pancreatic polypeptide is to self-regulate the pancreatic secretion activities, stimulate the gastric acid secretion, antagonistic of cholecystokinin and inhibit the pancreatic secretion which is stimulated by cholecystokinin. Excitation of the vagus nerve, administration of gastrin, secretin or cholecystokinin induces the secretion of pancreatic polypeptide. Neurotensin is a hormonal peptide of 13 amino acid residue present in the digestive tract as well as in the central nervous system. It acts as a central neurotransmitter or neuromodulator and a peripheral hormone. Potential actions of neurotensin include stimulation of colonic motility, inhibition of bowel and gastric motility, inhibition of gastric acid secretion and gastric emptying, stimulation of pancreatic, exocrine and intestinal secretions, increase of vascular permeability and vasodilation and thermoregulation. Neuropeptide is a 36 amino acid residue neurotransmitter secreted by the enteric nerve endings of ileum and column and also in medulla, hypothalamus and neurons of autonomic nervous system. It is a potent vasoconstrictor, increases blood vessels in enteric blood vessels, inhibits intestinal secretions, stimulates food intake and depresses the colonic motility. Bombesin is a 1,6,2,0-dipeptide of 14 amino acid found to be a potent stimulator of gastrin and gastric acid secretion, stimulates the release of motilin, cholecystokinin, neurotensin, enteroglucagon, vasoactive intestinal polypeptide and pancreatic enzyme secretions. It is also a potent chemoattractant of macrophages and lymphocytes and enhances the phagocytic process in macrophages. Bombesin also influences the calcium regulating hormones either directly or through its action on gut hormones. Willikinin, a peptide hormone, is released by the action of hydrochloric acid from the duodenal mucosa after a meal and stimulates the motility of microvillus. Guanylin, a 15 amino acid residue containing polypeptide, is secreted by the goblet cells and epithelial cells present in the colon. After the ingestion of a salty meal, guanylin is secreted into the intestinal lumen where they inhibit the sodium absorption and induce anion and water secretion. It regulates the electrolyte and water transport in intestinal and renal epithelia. Urogonylin is present in the enterochromaffin cells of the small intestine where it plays a role in regulating the pH after emptying stomach content. It plays a significant role in epithelial protection by increasing the pH of duodenal lumen by inhibition of hydrogen secretion and increase in bicarbonate secretion. The last hormone, which is leptin, known as satiety hormone, is made of adipose cells which helps to regulate energy balance by inhibiting hunger. It regulates appetite to achieve energy homeostasis and plays a crucial role in the adaptive response to starvation. Thus, this module helps to know the activities involved in generating the intestinal movements in different organs of digestive tract and also the needs of the hormone in all the activities. Thus, the gastrointestinal hormones produced in our body plays a vital role in the process of digestion and their activity are regulated by the digestive organs of our body. The gastrointestinal hormone regulates the secretory activities, homeostatic activities, absorption, phagocytic activities and so on of the digestive tract or the gastrointestinal tract.